All right. Hello, everyone. Welcome to VSPG. Today we have a presentation from Peter Crockford from Carleton University. And he's going to be talking about, or his talk is titled, How Much Life Has Ever Existed on Earth? Just a quick announcement before we get rolling. We accidentally scheduled a speaker for Thanksgiving. Here in the United States, I will not be hosting on Thanksgiving, so we've moved the Thanksgiving Day speaker to 2025, and we're still working on rescheduling him. So everybody take that day off in 2 weeks from now. Andre, anything else?
No. Yeah, so we continue until early December. I guess last talk is December 7?
First week of
No.
December. Yeah. First week of December,
Yeah,
the 5th.
and when we take a break and we come back in early January, and first couple talks will be actually in later time because we will have speakers from Australia, but we will announce it.
All right, great. Thanks. So Andre, please go ahead and introduce Peter for us.
So it's a pleasure to introduce Peter. Peter did his both undergraduate and graduate in Canada. He is originally from Canada and did his undergraduate in University of Victoria in BC and PhD at McGill University, working with both Wynne and Galen Halverson on sort of Precambrian biogeochemistry and combining like sedimentology stratigraphy and isotope geochemistry. He's particularly interested in multiple sulfur and oxygen isotope. That was a big part of his PhD, well, among other aspects. And he followed with a postdoc between Weizmann University and Harvard University, working with David Johnson and Itai Halevi, and when another postdoc at Woods Hole Oceanographical Institute, working on barium isotopes. And he will talk today about sort of how using multiple oxygen isotopes, it's possible to constrain productivity on early Earth. So with this, I pass it to Peter.
Great. Well, thank you so much for the invitation. And thanks so much to you and Alex, Andre, for putting this all together. It's been a huge resource to both me and students. So today I'm going to talk about work that, like Andre mentioned, started through my PhD at McGill and has sort of been a consistent theme throughout my early research career. And what excites me about this topic is I think that these new geochemical tools that are coming online give us new windows into the carbon cycle's evolution through time. And that's going to be the focus of the talk today. So the overall focus of my work and my group's work has largely been to hopefully refine cartoons like this of Earth's past. So this is a figure from a paper I published in 2019 with a number of collaborators. And even though these broad trends appear to be true, CO2 has decreased steadily throughout Earth history as it's compensated for the faint young sun. This two-step rise in O2 has, I guess, first proposed many decades ago, but is continually being refined by the community, and how this projects onto the fascinating history of life that's emerging from molecular clock records and the fossil records with transitions from prokaryotes to eukaryotes and eventually animals. The goal is to really refine this sort of picture and understand how it translates into the marine environment where we have access to much more material to analyze for our studies. So thinking about how can we constrain things like pH over time, marine redox conditions, and then use that to inform on atmospheric evolution and the evolution of the biosphere. So The route that I and many who have been received a similar training as I have focused on this is by both trying to find lessons from the modern environment and develop geochemical tools that we can then apply to the geologic record. And hopefully with the care that and knowledge that the Earth has considerably changed over its 4.567 billion year old history. And the way that we build this bridge is largely through the sedimentary record. And so just some photos on the bottom part of the slide here from a couple from Baffin Island. And then on the far right, some evaporates from the Mackenzie Mountains. This has largely been the focus of mine and my group's work. So one example, I think, of a record that really shapes many of our views of Earth's past is the stable carbon isotope record in carbonates. And all I want you to draw your eyes to is that this record has quite a bit of variability. And a lot of work has been done to try to understand exactly what is driving this. So at the beginning of the Proterozoic, around 2.4 billion years ago, we start to see this precipitous rise in carbon isotope values. And this largely underlies models of Earth's great oxidation event. And then later in the Neoproterozoic, we see very large negative carbon isotope excursions, which have also been interpreted as large perturbations to the global carbon cycle. But there's been considerable debate about what this record really means, how we should go forward in interpreting it. But I think that there's some very basic questions that sort of drive much of my interest that gets at why I'm interested in working on problems like that, which we can reframe into some very, very basic things that I think would be appropriate to sort of pose to folks in elementary school or high school, which is how much carbon has ever been fixed by the biosphere? How many cells have ever existed on Earth? What has been the most abundant type of organism to have ever existed? What impact have extinctions had on Earth's biospheric history? And where is the majority of the uncertainty that could help us refine this record of Earth's past? Now, my main focus today is going to be on primary productivity. So I'm sure, as many of you know, there's a lot of organisms that we could think of as primary producers, utilizing a number of different metabolisms. But by far the most dominant one on Earth today is oxygenic photosynthesis. So this is a conversion of inorganic carbon to organic carbon with the important byproduct of O2. And now today, this is the combined efforts of marine microorganisms such as algae, cyanobacteria, and on land, it's largely due to terrestrial plants. And today, about 45% of this is performed in the ocean and about 55% on land. These numbers are constantly being refined, and they've likely varied a fair bit throughout Earth's past. Now, today, collectively, this results in about 250 billion tons of carbon fixed through this reaction per year. And of course, without this, we wouldn't see any of the diversity of life we see on the planet, forming the basis of the food chain. Now, I think as we look throughout Earth's past, there is some intuition of how the biosphere may have evolved. So on the lower right hand of the slide, what you see is basically a census of the biosphere, just looking at dry carbon. So you can imagine that if we basically kill everything on Earth, remove all the water, and just put the dry carbon on a scale, where does most of it reside? And of course, plants are the largest portion here. You can imagine trees are big, they weigh a lot. What might be less intuitive is the large contribution from bacteria. That's because they can occupy so many niches and extend fairly deep into the Earth's underneath the Earth's surface, which ends up being a huge amount of material. And things like us, animals, all of our livestock, pets, and wild animals is the lower right corner with just a very, very small contribution. But as we go back in time and we look at the fossil record and the molecular clock record, can we simply calve off chunks of this figure? Can we simply remove land plants, animals, fungi, this sort of thing? And does that mean that we had a much smaller biosphere in Earth's past? And so I got very excited about this paper, which was presented by Inon Baron and colleagues Ron Milo at the Weizmann Institute. And that ultimately led to thinking about some of the questions I posed at the start of the talk for a fun postdoc project during COVID, which I'll largely focus on today. So can we think of this sort of record when we remove chunks of the biosphere as simply less life on Earth as we go back in time? So if we can constrain productivity of the biosphere, which is really the base of the food chain and drives a lot of this, can we actually get to some of these fundamental questions which I posed at the beginning of the talk? Now, measuring productivity today is done through the combined efforts of satellite observations, shipboard observations, looking at bottle incubations and extending this over broader swaths of the oceans. We have forest observatories, and it's been a pretty amazing accomplishment, I think, from a lot of the community that's put this together, that we can really measure sort of the biosphere's contribution to so many of Earth's biogeochemical cycles, but paying special attention to just the rate of primary production any given day or any given year. But you can imagine that if we were to sink all of our ships, get rid of all of our forest observatories, and crash all of our satellites, this becomes a very, very difficult problem to solve as we go back through Earth history. Now, one place we can look is the atmosphere. So as pointed out by the earliest note of this that I found is James Lovelock in the 60s, but you can find breadcrumbs which probably led to this idea much earlier, is that the atmosphere is a reflection of a living biosphere, especially on Earth today. And a clear example of that is just looking at methane and O2. If we were to kill off all life on the planet today, or at least accelerate current efforts, O2 would slowly oxidize, reducing material that's constantly being resurfaced on the continents, to a lesser degree in the marine environment, and would eventually be drawn down. Methane would be oxidized much faster, and we wouldn't really see this sort of disequilibrium in a dead planet, especially not to the degree that we observe on Earth today. Now, what I focused on is just looking at the isotopic composition of O2 and how that may have evolved through time. So oxygen has 3 isotopes, 16, 17, and 18, but in order of abundance, 16, 18, 17. We represent this in delta notation. So we're working with whole numbers. That's why there's that multiplication by 1,000, and also reference to a standard. So 0 means the same thing to everyone. And it's usually with the rarer isotope over the more common one. So 18 over 16 or 17 over 16. Now, if we take these two properties, delta 18O and delta 17O, and plot those like I have here, essentially all Earth materials will fall along a straight line, because most processes, the vast majority of processes on Earth, follow a mass-dependent behavior, where an isotopic substitution of a 16 for a 17 will be half of the isotopic impact of an isotopic substitution of a 16 for an 18. Now, when people started making these measurements, these sorts of relationships really demotivate one to over-engineer a mass spectrometer, because if there's no new information gained from measuring the rare isotope, why would somebody go about that, since we get everything we need to know from just looking at oxygen 18 measurements? That was until the 1970s out of Bob Clayton's lab, where a second set of materials was measured that did not seem to follow this mass dependent behavior like I just showed you. And they seem to follow a slope of 1, meaning there was no mass discrimination between a 17 or an 18 following this sort of mass-dependent relationship. Now, the important thing about these samples that are plotted here is that they're actually meteorite samples. And so an early interpretation of this was perhaps these are oxygens from a different reservoir than we're dealing with on Earth that would have a different distribution, and that might explain this data set. But a postdoc out of that lab, Mark Tiemens, I believe when he started his job at UC San Diego, performed a series of really important experiments, where what he did is he took O2, started to spark it to produce ozone, and measured the oxygen 17 and 18 composition of the product ozone and the residual O2. And what was found was that over time, the ozone becomes enriched in oxygen 17, and the O2 is left depleted in oxygen 17. Importantly, this seemed to be a large departure from mass-dependent behavior and seemed to follow a mass independent behavior. And the important contribution here was that this seemed to give rise to a new type of isotope effect, a so-called mass-independent isotope effect, which has largely been the foundation of much of the work I've done subsequently, and many people have. So the way that we define this mass independence is simply a deviation from mass dependence. So if we have our measured oxygen 17 value, we simply subtract what we would have expected that value should have been from pure mass dependence. And throughout the talk, I'll frequently refer to this as either triple oxygen isotope value, which is this capital delta 17O symbol here, mass independent, or OMIF. So I apologize if I sort of interchange those terms. Now, knowing that there was more information to be gained through making these triple oxygen isotope measurements, people started going out and measuring these in the environment. And looking at just atmospheric constituents, folks started measuring ozone, nitrate, CO2, peroxide, sulfate, and importantly, all of these atmospheric species seem to bear a significant mass-independent enrichment in oxygen 17, or a positive cap-17 signal. Now, if all of these gases are plotting positive or have an excess of O17, something has to be bearing the brunt of a negative signal for mass balance. And of course, that's O2. So again, I've just plotted the magnitude of these signals again, just on a simple plot here with oxygen 17 values on the x-axis and O3, CO2, and O2. And you can see extreme isotopic values with O3 having a triple oxygen isotope value of up to 40 per mil almost in the modern atmosphere, CO2 up to something like 12 or 13 per mil, and O2 just carries a small negative signature. And the reason that it can carry such a small negative signature and make up for all of those very, very enriched values in O3 and CO2 is because there's so much more O2 in the atmosphere than these other constituents. Now, thinking more mechanistically about what's going on here, what's actually driving the magnitude of these changes between these different species, folks like Yuck Young out of Caltech started to do put together some models to constrain this. And an important insight from that work was this relationship between the magnitude of triple oxygen isotope depletion in O2 and how much CO2 is in the atmosphere. Later laboratory work seemed to confirm this inference. This is, again, experiments out of Mark Tiemann's lab, where you see with increasing CO2, we get increasingly more negative triple oxygen isotope values in O2. Now, moving beyond the laboratory into the natural environment, people started to measure this in ice core samples. And what we're looking at in the plot in the middle of the screen there is our triple oxygen isotope value of O2 and versus CO2 concentration. So you can imagine that we're taking O2 and CO2 from these gas bubbles, in this case in the Gisby ice core in Greenland, and we're measuring the triple oxygen isotope composition of the O2. And what you can see is that with lower CO2 levels, we have a less negative triple oxygen isotope value, and with higher CO2 levels, we have a more negative triple oxygen isotope value. Or another way you could think of this is at the top left, we're in the last glacial, and in the bottom right, we're in the interglacial. So all of that intro was just to say there's a clear relationship in the magnitude of the triple oxygen isotope signatures that we see and how much O2 and CO2 are in the atmosphere. So if it was as simple as that, we would have essentially a perfect CO2 barometer that we could take through Earth history, but it's a little bit more complicated than that. So in the stratosphere, we have these photochemical reactions that are driving this mass-independent behavior that's spread out between O2 and CO2 and other gases, which leads to a very, very negative value in atmospheric oxygen. So that comes down from the stratosphere into the troposphere. But of course, there's another important component that we need to think about, which is the biological flux of O2, which sources its oxygens from water, which carries no mass-independent signature. So, and of course, because photosynthesis is by far the most dominant metabolism on Earth today, driving primary production, we can approximate this O2 flux as the gross biospheric productivity or gross primary productivity of the biosphere. So it's really the inception of these signals through these stratospheric reactions and the constant erasure of biologically produced O2, which sets the tropospheric composition that we can go out and measure today. So some predictions for this system are that, one, we should not see an appreciable triple oxygen isotope signature in atmospheres with no oxygen. So on Earth, perhaps we could directly test this in the Archean. Two, if we can constrain how much O2, CO2, and primary or O2, CO2 or primary production, if O2, CO2, and primary production have changed throughout Earth's past, then this should be reflected in changes in the triple oxygen isotope record of atmospheric O2. And third, if we know 2 out of 3 of these variables, we should be able to solve for the third. So we run into a quick problem with this system is that the ice core record only extends back at most about 6 million years. So this is new results that have come out of really incredible work by a large research consortium. Folks at Princeton who I've worked with, like John Higgins, have been, I think, leading such efforts. But these are unhinged ice blocks sort of at the margins of Antarctica. And the longest complete records, I believe, extend back around 800,000 years. But if we're interested in things like biospheric change over all of Earth history, we need another record of oxygen. So the place that myself and many colleagues have focused is sulfate. So you can imagine that as this stratospherically photochemically reacted oxygen gets mixed into the troposphere, mixes with that biospheric O2, some of that oxygen will react with reduced sulfur in things like terrestrial environments, oxidizing minerals like pyrite, and a product of that reaction is sulfate. And some laboratory experiments have constrained the amount of oxygen incorporated into that sulfate to be around 8 to 15% of the oxygens in product sulfate derived from O2. Importantly, because the rest of those oxygens come from water, which does not carry any mass-independent signature, we can think of sulfate as a diluted version of our O2 triple oxygen isotope value. So putting all of that together, we have stratospheric reactions that produce very, very negative triple oxygen isotope values. That mixes into the troposphere with a biological flux of O2, which sets the tropospheric composition. Some of that O2 can then oxidize sulfides to produce sulfate, which we can think of as a diluted version of our atmospheric O2. And if that sulfate can be spared biological sulfur cycling or microbial sulfur cycling, perhaps that sulfate can retain that isotopic composition and be deposited in the geologic record for us to study. So to build up a record, I've been extremely fortunate to get to go to some pretty wonderful places around the world, largely in the Canadian Arctic, but a little bit in Australia and in places like California and Spain. And I've relied on extremely generous collaborators. In particular, Andre has been very generous with providing materials so we can try to populate as dense of a record as possible. Just some pictures just to show some of the diversity of some of these environments. Here we're in the Angmat Formation of northern Baffin Island. And what we found within this, which is just a carbonate-dominated unit, is little pods and thin layers of gypsum, which I got quite excited about. Now, I'm not a huge fan of heights, so you can imagine this is sort of intense field work for me. But if you look at my colleague, Marcus Kunzman here, who's just at the center of the photo, we'll see him now again at the lower left of this picture. And now Marcus is sitting at the base of what's called the Tenstone Formation in the Mackenzie Mountains. And I'm holding the camera here. And what Marcus is sitting at the base of is about 100 meters of thick bedded gypsum. Okay, so we can get very, very different expressions of these units in sedimentary record. A third example, or third and fourth examples, are looking at barytes. So on the top, we have barite from Brazil during deposited at the aftermath of the Marinoan glaciation, okay, with this interesting fanning textures. And in the bottom, we have barite from the Belcher Islands, okay, in Hudson's Bay, Canada. And we have these centimeter-sized barite crystals in the Quesagilic Formation. Okay, so putting all of the data together, this is, of course, not just an effort by myself. This is a compilation of basically everything I've been able to find through the community. But when we plot this up, we can look at how seawater, rivers, but then in geologic materials, barite, carbonate-associated sulfate, iron oxides, and gypsum have varied throughout time. And so to date, as best as I can tell, there's about 650 measurements made, and that number keeps growing. So just to put some important constraints on this figure, where we have triple oxygen isotope values in sulfate on the y-axis and the history of the Earth from about 4 billion years ago to present on the x-axis, at about 0, around that 0 per mil, we have modern marine sulfate, and around minus 0.5 per mil, we have modern tropospheric O2. Okay. Now we can break this up into specific types of sulfate. So what I've highlighted here is just gypsum and to a lesser degree anhydrites, which dominates most of these measurements. There's been a much more limited amount of carbonate-associated sulfate measured. The simple reason for that is it's a fair bit more labor intensive. And so that's certainly an area that I think requires a lot more attention, and I think we could learn a lot from it. And I'll come back to this at the end of the talk. We have barites, which offer us some of our earliest sulfates in the geologic record. And so the goal of this talk is going to be walk you through this record here through 5 fairly fast little vignettes. Just to begin, your eyes might be drawn to what's happening here at about 635 million years ago. Just briefly, this discovery was initially made by Huiming Bao in 2008, although he made these measurements much, much earlier, and I think sat on them because maybe they were a little bit difficult to understand initially. But this was an incredibly important contribution because at the time this was published, I believe there was a lot of debate around whether the Marinoan glaciation represented a hard snowball or a slush ball. And the key takeaway from the study of Huiming was that the way that you would generate such large signals would be an incredible amount of CO2 built up in the atmosphere. And it's difficult to understand how one would do that in a slush ball scenario. And this seemed to provide a sort of smoking gun for a hard snowball scenario. And so it was an incredibly impactful study and something that actually got me interested in this field in the first place. And so the idea is that during a snowball Earth glaciation, once you extremely limit air-sea and sea-land or air-land gas exchange, you could build up extremely large concentrations of CO2, which would ultimately be required to tip the Earth out of a panglacial state and deglaciate the planet. And I think one of the coolest things with this, these records where we've measured these barites around the world is that because this is tied to the atmosphere, I think that these signals in units distributed around the world provide a very interesting geochemical datum that can tie these units together in time. Going back in time, looking around Earth's great oxidation event, we can plot up triple oxygen isotope values, just looking at values in red here for sulfates deposited before the GOE. So this is largely reliant on the barites from the North Pole Dome and Barberton, and compare that to sulfate units deposited across the early Paleoproterozoic. And the important thing is that there is an increase in the magnitude of the triple oxygen isotope anomaly as we go into the Paleoproterozoic. One way that we could interpret this is that this could be the inception of an ozone layer. In early models out of Jim Casting's group, I believe, by Pavlov, constrain this to about 1/1000th of the modern inventory of O2. So if we look at, for example, reconstruction of O2 through time out of Tim Lyons, Noah Planowski's, and Chris Reinhardt's review paper from 2014, we can make a little bit of progress by suggesting that maybe we can put a lower limit of O2 at about 10 to the minus 3 over basically ever since the Great Oxidation Event. Okay, looking at the mid-Proterozoic, I'm largely going to focus on the Sibley group. Okay, so the reason for working here was one, there was available material. This area is prospective for uranium exploration, and so a number of drill cores were available. This is samples provided by Phil Fralick. And second, this is interpreted as an ancient lake environment. And the thinking behind that is that in ancient terrestrial environments without a terrestrial biosphere, that would perhaps limit organic matter input into such environments, which would limit things like microbial sulfate reduction, which may give us the best chance of preserving triple oxygen isotope signatures in the sedimentary record. Okay, so again, going back to that simple schematic of what I presented at the start of the talk, now we have our sulfate samples. Now we want to project back to what the value of tropospheric O2 would be. So we're just going to undilute that atmospheric component of these samples. Okay, and the goal is to try to pull out what the biological flux of O2 would have to be to make these samples make sense. Okay, so I'm not going to walk through all of this, but just one case, one property that there's a lot of debate about. Just zooming in on O2, there's huge amounts of debate over what O2 levels were over the mid-Proterozoic. Are certain geochemical records that suggest very low O2 values? Are those recording true baselines? And are all of these transient excursions people have started to point out with things like iodine-calcium ratios, are those small transient events? Or perhaps are some of these small transient events actually recording the true values, and we have to worry about things like diagenesis? But the point is that there's a huge amount of controversy in this question, but we can walk our way to a slightly narrower range. So because of sulfur isotopes that there's been a number of VSPG talks on, we can constrain O2 levels to at least be above 1/100,000th of the modern inventory, or about 10 to the minus 5. Okay. At the high O2 end, we can say that if we look at marine records, we see a very heterogeneous mix of ferruginous and euxinic conditions and shallowly oxygenated conditions. And we can ask the question of how much oxygen do we need to basically fully oxygenate the oceans? And so if we find this heterogeneous record, we must be below that threshold. Going back to the lower end, work by folks like Colin Goldblatt have put forward that due to the dynamics of things like methane and O2, that the atmosphere is fairly unstable in very, very low O2 conditions, from about 10 to the minus 5 to about 10 to the minus 3. And like I mentioned earlier, we can say because we have a triple oxygen isotope signature from the GOE onwards, that O2 is likely higher than about 10 to the minus 3. But it's really debate within this 10% to 0.1%, or 10 to the minus 3 to 10 to the minus 1, if we want to say it, that I think exists in the community today. Now, if we combine features like that with photochemical experiments, which have done all the isotopic accounting of how oxygen isotopes would vary under different O2 and CO2 conditions, and we put that into a simple model, simply saying, if we know what CO2 and O2 are, we can constrain what the stratospheric value would be. And because we have our triple oxygen isotope value in sulfate, we can constrain what the tropospheric value would be. And we can simply say, what's the biological flux of O2 we need to make those two properties make sense? Okay, and the approach we took for this was a simple Monte Carlo approach. Now, if we go back to that high, thinking about the debate of O2, whether it's high O2 in the mid-Proterozoic or low O2 in the mid-Proterozoic, we can actually see the biospheric consequences of those two different models. So in the red histogram here, we have our estimates for a low O2 world. So from about 0.1% modern O2 to 1% modern O2. And that gives us an estimate of a biosphere that's about 6% as productive as the modern biosphere. For the high O2 case, going from about 1 to 10% of modern O2 values, we estimate that about it suggests about 41% of modern biological productivity. Now, this describes two very different planets. In the high O2, high productivity world, it essentially suggests that the Earth back at this time was operating at modern marine productivity levels. So you can imagine we get rid of the terrestrial biosphere and we're in a Proterozoic-like state. In the low O2, low productivity case, it suggests there are other important mechanisms at play. So could this be metal limitation in the ocean is leading to nitrogen limitation because maybe organisms have a tough time synthesizing nitrogenase? Could this be a reduction in phosphorus inputs due to less oxidative weathering and some important feedbacks with things like the iron cycle? Could it be due to a less complex biosphere, perhaps less nutrients from less weathering just overall? But it would suggest that we would have about 15% modern marine production. At the moment, I would say that we can't conclusively rule out either one of these scenarios. I tend to favor the lower O2 case, but I would be excited to be corrected on that. Okay, moving a little further back to what I would consider the beginning of the mid-Proterozoic, or sort of the end of Earth's great oxidation event, as defined by folks like Dick Holland, we see a really steep decline in triple oxygen isotope values. Now, these are results from the Belcher Islands. So for those of you who haven't played around on Google Maps and looked at this, images this from space, I think it's one of the most interesting looking island archipelagos in the world. And what's housed here is a rare glimpse into the Orosirian through a nice shallow marine depositional environment. And this was field work done by my colleague Malcolm Hodgkiss, which started in his time as McGill, and then he largely focused on for his PhD with Eric Sperling at Stanford. Again, spectacular place. Malcolm was able to constrain the age of the lower part of the stratigraphy here, the Cascillic Formation, to be around 2 billion years old, and importantly found these barytes within these units. Now, thinking about how one could interpret this large drop in triple oxygen isotope values going from a baseline of around -0.3 all the way to about -0.7, I think there's two hypotheses one could take. One is we just have a reduction in the amount of atmospheric oxygen that is making its way into sulfate. And so this could be due to changes in the amount of microbial sulfate reduction operating in these environments, changes in oxidation mechanisms. I find this a little harder to digest because we're sampling across a range of different formations. It's strange to think that there would be a temporal difference between different time intervals based on such local factors. And it also ignores us to ignore a lot of geochemical data speaking to changes in O2 across this interval of time. And so also, I don't think like local factors like MSR would operate so differently in space and time like this. An alternative interpretation is that we are seeing real changes in the surface environment. And one way that we can interpret this, if we also take into account estimates of O2 and particularly rely on some of the upper estimates of O2 for studies that have reported evidence of an oxygen overshoot, this could suggest that we see a decrease in productivity by as much as 100-fold across this interval of time. Even if we hold O2 constant and just look at the triple oxygen isotope results and say, what would they infer for a change in productivity? We get a reduction in about 5-fold, or about 80% of the biosphere gets wiped off the planet. And of course, there's a large degree of uncertainty with exactly how much time we're talking about, but we can confidently say that this probably occurred in much less than about 40 million years. Again, this isn't an extinction because we're only dealing with the microbial world, but it does seem to be indicative of a fairly large loss of life. Okay. If we try to take these records and actually apply the rationale I had to the entire record of sedimentary sulfates throughout the whole geologic record, and put it through this framework that I just mentioned to you, and try to constrain the isotopic value of tropospheric O2 for all of these time points through Earth history and try to pull out what this biological flux of O2 would be through time, we can come up with a record of the productivity of Earth's biosphere through all of Earth history. Now, of course, the upper panel here is actually the result of a combination of a number of studies that have gotten at productivity, whether it's through models constraining nutrient levels and then calculating an inferred primary production, other geochemical tools, and it's cobbling all of those together and asking, one, is there agreement between all of these different ways of getting at this problem? And two, how much uncertainty is there on this question through time? So the important takeaways is that when we combine all of these records, it does seem to show a sustained increase in productivity throughout Earth's history, with notable jumps and important plateaus as we walk through Earth's past. But the other thing to note is that there is considerable uncertainty in these estimates, which should be certainly something that motivates me and my group to hopefully refine as we move forward. The other thing I want to draw your attention to is that there's a huge amount of uncertainty as we move into Earth's great oxidation event. And this is largely a result of ideas about an oxygen overshoot. So as we move above, if we look at sulfur isotopes, it suggests that we moved above about 10 to the minus 5 present atmospheric levels of O2. But how much higher did we go? And our proxies for how high O2 could have been are considerably more uncertain. And so if we had extremely high O2, we generate extremely high productivity estimates. If we had much lower O2, it becomes much more muted. Now, if we project this productivity picture we see above, and we match that onto what metabolisms and organisms that we imagine and have evidence for inhabiting the Earth at these different times, we can come up with a very, very interesting history of the Earth and answer some of the questions that I posed at the beginning of this talk. So for example, how much carbon has ever been fixed? How many cells have ever existed? What has been the most abundant type of organism to have ever existed? What impacts have extinctions had? And where's the majority of the uncertainty that as a community, it would be, I think, fruitful to really focus in on? We can provide answers to these questions. So how much carbon has ever been fixed? We can estimate about 10 to the 11 to 10 to the 12 billion tons of carbon ever since the origin of life. That's a pretty large value that many of you probably don't think about a lot in your day-to-day life. Just to give an example, if the Earth was completely composed of carbon, or sorry, if the moon was completely composed of carbon, and you look at it tonight, that would be about 10 to the 11 billion tons of carbon. Okay. How many cells have ever existed? We can estimate that about 10 to the 39 to 10 to the 40 have ever existed to date. And we can even project that forward in time and put forward that Earth is likely to only ever house about less than 10 to the 41 cells ever. Of course, that's in the absence of us being able to geoengineer our way out of when the sun eventually goes into a red giant and things like that, or multiply the productivity of the biosphere by multiple orders of magnitude through future generations' ingenuity. This is just assuming if we roll the clock forward and think about when CO2 drawdown is going to start to really limit the terrestrial biosphere, and eventually the oceans will get too hot for the marine biosphere. What has been the most abundant type of organism to have existed? We can say with some confidence it's been cyanobacteria. What impact have extinctions had on this biological history? We can argue perhaps none, but we could maybe even suggest that perhaps it's overall led to an increase, because although there is a transient extreme decline when extinction events have hit, the innovation that's been occurred after these events may have led to an overall increase in life over the long term. And where is the majority uncertainty? From my point of view, I think it's around Earth's great oxidation event. So I welcome you to check out this work in a recently published paper in Current Biology last fall. Now, I think as a goal, it should be to try to refine pictures like this as we move forward. So I just want to zoom in here on the mid-Proterozoic. So if you don't take away anything else from this talk, one thing I just want to draw your attention to is that we have values in sulfate that are more negative than modern tropospheric O2. We cannot generate sulfates on Earth today with values like this. Okay, so if you one takeaway I'd hope you take from this is that these sulfate signals have to be telling us that the Earth was fundamentally different in Earth's past than it is today. Okay, but I also want to draw your attention to there's considerable variation in triple oxygen isotope values if we look at any individual formation or broad swath of Earth's past. So what's driving that variation? Is it real-time environmental change, or is there other factors at play? So one thing I've worked on, started my postdoc, and we're hoping to submit this shortly, is just looking at the different types of depositional environments these sulfates have found themselves in. Okay, and if we split up that mid-Proterozoic record into sulfates that we can confidently assign a terrestrial origin, sulfates that we can say are extremely restricted environments or semi-restricted sort of peritidal environments, or sulfates that are in much less restricted settings, or even looking at hydrothermal barytes, which should just be sourcing sulfate from pure seawater. And when we look at the magnitude of the triple oxygen isotope signal between these environments, we see a notable decline basically all the way up to values that we couldn't distinguish from modern values. And so I think what this shows is an extremely strong basis control, which means that the depositional environments that are preserving these sulfates, we need to be include this sort of thinking when we look at the variation in the geologic record. And so when we have variation within an individual formation, is that real-time environmental change, or is it local basinal factors driving that variability? I think it's almost certainly local basinal factors for the most part. Now, the other thing that I think is important with this is it seems to show a strong, we seem to have a strong signal for terrestrially influenced environments. And I think this could be very, very useful as we look forward. So if we take upon ourselves, if we think about the framework that was initially put forward by Huiming Bao, where we have terrestrially oxidized pyrites, which produces sulfate that eventually makes its way into the marine environment, and that along this transport from initial oxidation all the way to moving into the marine realm, we're actually losing this signal. I think the data set that I just showed you makes a ton of sense. Now, coming back to the carbon isotope record, where there's this considerable debate around the nature of this record, not just the carbon cycle models we apply if we're interpreting this as purely global changes in the carbon cycle, but even what is the degree of diagenetic alteration and how is that affecting the record we see today, but also how many of these units don't belong in such a compilation of global seawater and have been extremely influenced by the terrestrial environment, perhaps overprinting some of these signals, or carbonates that had no connection to the global ocean whatsoever. And I think things like carbonate-associated sulfate may offer a powerful tool in helping with that, thinking that if we have large signatures preserved in our carbonates, perhaps that means that we're much more terrestrially influenced, and therefore perhaps we don't include these samples in sort of global compilations. And so again, the cast record is extremely sparse, but hopefully in the coming years that will be alleviated. And I know this interpretation departs from some of the recent work that Huiming Bao has put out, thinking about oxidation of sulfide within the water column, which I think is perfectly valid and very interesting idea. But I would argue from the data that I just presented, I think his original model of terrestrial pyrite oxidation seems to support that data set quite a bit better. The last thing I'll just touch on here is work by Anna Waldeck, formerly out of Dave Johnson's group, now at Penn State, who did some really interesting work on the Paleozoic. Now, the rise of land plants is something that's very controversial. If we go back to just sort of the insights from folks like Senior and Lips from the 1980s, just talking about the imperfections of the sedimentary record, obviously the earliest forest occurrences we see are obviously not the first forest that ever existed on Earth. And so that leads to some uncertainty exactly when the first forests arose on Earth, and also in general, just the rise of land plants and that timeline in general. Now, looking at the triple oxygen isotope record throughout the Paleozoic, there's a notable rise in triple oxygen isotope values. Okay, now thinking about what I just mentioned with this strong depositional environment control, if we're going to be comparing true environmental change through time, we should only really be comparing apples to apples, or providing some sort of correction between depositional environments. In this case, we're only sampling marine evaporites. Okay, so you could think of analogies like the Messinian gypsum, stuff like that. And if we look at this sort of record, if we just look at sulfates before about 407 million years ago to about 480 million years ago, and sulfates deposited after from about 387 million years ago to 308 million years ago, we see a marked increase in triple oxygen isotope values. Now, could this be an increase in productivity, O2, a decrease in CO2, a decrease in the amount of oxygen getting into sulfate during pyrite oxidation because of changes in the weathering environment imposed by the rise of land plants? I would argue all of these are consistent with the rise of land plants, which I think is a step forward in thinking about when did the rise of land plants become an extremely important force in driving Earth's biogeochemical cycles. So that's all I've got for you today. So thank you so much for your attention, and I'd be happy to answer any questions. Thanks.
Thanks, Peter. That was great. We're going to move to the question and discussion section. And you can ask a question a couple ways. You can raise your hand by finding the reactions button at the bottom of your screen, and then there's a raised hand button there. Or you can type it into the chat and I can ask it for you. So your first one is from Greg Rotalic. Please go ahead, Greg.
Oh, Peter, that was inspiring. And it agrees entirely with the Paleosol record, because I have productivity increases from three aspects of the Paleosol record: base depletion in the Paleosols, from depth of salts in the Paleosols. And Ilya Binderman and I just published the isotopic difference between Paleosols and the ocean in scientific reports. So we're really on the same page here. But I have a question, and my question is, a lot of the sulfate in the Archean, I think, is not a simple marine evaporite reaction inorganically with oxygen. It's created by sulfur oxidizing bacteria. So my question is, what effect does that have on Cap Delta?
No, I mean, for the Archean record, we have so little to go off of. And so you're right, like we got to be careful not to overinterpret it. For the North Pole Dome, for example, it's not really interpreted to be, latest I've seen, it's not interpreted to be a marine environment at all. In terms of specific mechanisms within the triple oxygen isotope values we see coupled to the sulfur, that's a really, really tough thing to deconvolve, because it's difficult to pull out effects we see in the lab today and interpret those signals in the backdrop of all the independent signatures that we see as well. So that's a little harder. The Paleosol stuff, that's interesting. And yeah, as soon as teaching winds down in the semester, I'll start digging into that and check that out. So thanks for bringing my attention to that.
Well, my question is basically that there must be a Rubisco effect or some enzymatic effect in the oxidation of pyrite by bacteria. If for no other reason than the ordinary delta carbon for cyanobacteria is usually about -32 to the -35. So it's a little bit more negative, more extreme than you find typically in cyanobacteria, which is typically -25 to -28. So there must be something there. If only if you find some evidence, or I'd be really interested to know.
Yeah, thanks. Yeah, I'll think about it. Thank you.
Brilliant. Love it. Thank you.
All right, thanks, Greg. Next was Jingjian. Please go ahead.
Hi, Pete. That was a great talk. For a short question of
Sure.
how did you derive your total number of cell number, or what kind of assumption went into that, like according to the GPP estimate?
Yeah, no, thanks for your question. I actually, when I was thinking about putting together this talk, my bet was that you'd be the first person to ask a question, actually. So the way we did that was like looking at current cell populations estimates. So there's been a lot of work on this, thinking about when we have a number of primary producers in surface oceans, how many heterotrophs does that translate to, and work our way all the way down to the seafloor. So even though the most cells today actually occupy like the deep subsurface and stuff, their turnover times are so slow that you can actually gain most of your needed information just by looking at sort of the upper parts of the water column. And so we relied on those relationships to extract our primary productivity values into total cell numbers. And when you start removing things like trees, animals, all that stuff, they end up being a very, very small rounding error in that. It's really the primary producers and sort of the first set of heterotrophs that really drive a lot of that cellular turnover, which drives the estimates we have here.
Yeah, a quick follow-up. I guess my question is how you kind of integrate over time, right? Like this, you are right, the surface oceans, well, the turnover time, the microorganism in the surface oceans is very short, right? And we're dealing with billions of years. So do we have any kind of constraint on how you can do that?
I would say that we have, so we have the modern constraints of cellular turnover times that we were able to, that's what we applied to this study. But if you were to ask me how much error should we put on that number, thinking about surface seawater temperatures, the organisms that inhabit it, I'm not sure. That would be something that would probably increase a little bit of the uncertainty. I don't think a whole lot, though.
Yeah, but Pete, like your like framework was amazing. Like even if there's a great uncertainty of the turnover time, your number will still put an upper limit on the total amount of biomass, which is I think that's fabulous. Thank you.
Yeah, and I think when we get up to numbers like 10 to the 40, that a lot of the uncertainties that are, we're talking about don't really come up. But thanks for your question. Thanks.
All right. The next one, I'm gonna hit one from the chat from Rob Rainbird. Have you looked at abundance of organic rich shales over time as a proxy for organic productivity, taking into account sedimentary recycling, which would affect older strata?
No. I would have obvious problem I would see with going down that route would be if we look at times like the Loma Gundy, which is like largely gives us the basis for a lot of the arguments for the auction overshoot. It's like if we don't have the rocks to actually constrain that, and being cognizant of things going through the oil window, and I think we might end up with a record that kind of leads us a little bit astray. I'm not sure it would actually be productivity, but maybe just a record of black shale preservation. It'd be interesting to look at. I think some people involved in the sedimentary geochemistry paleoenvironments project are looking at this. I'm curious how it maps on to records like this, which would be interesting. But yeah, that's sort of the best I can give you with that.
All right, thanks, Peter. Next, Francis McDonald, please go ahead.
Hey, really nice talk. I enjoyed that. So Huiming Bao, since his work on the Marinoan cap carbonates, many of us have interpreted the signal from the barytes in the cap carbonates as being a signal of high CO2. But Saint-Geoffrey et al. Made the argument that, hey, maybe this is a record of low primary productivity instead. And I think, after your paper making an interpretation for the middle Proterozoic for those 17 signal as low productivity, I was wondering if you could just comment on going back to those interpretations and why you choose to still interpret the barytes in the cap carbonates as being a high CO2 signal rather than a low productivity signal.
Yeah, so I think part of it has to do with like where the barytes are in the stratigraphy. So because they're like not at the absolute base or deposit like during the glaciation, that I think that gets a little bit harder to.
But I was just going to say, but the CAS records, right? The biggest excursion is in the
Yeah.
diamictite actually, and they
No,
decrease
I
the
know.
convection.
Yeah, so obviously, like the way I've set things up, of course, there's a balance between all these processes that could be driving this signal. For the barytes, though, I think for where they're deposited and that you would likely have, I don't know exactly how many years, there's debate on that, enough time, I think it still makes more sense to me that it's still a remnant of a high CO2 atmosphere than depressed productivity.
But if the largest signal is in the glacial rocks and we
So
just
I'm saying
get
it
the
could be,
barytes when we happen to mix, then why aren't the barytes just recording that glacial signal and we're just seeing the mixture up top? It's just when we happen to record the barytes.
well, I guess the way that I've envisioned it is that you turn on productivity after your glaciation. That would be helping increase O2. That eventually would be pushing to overprint. That would turn on quite quickly after the glaciation. And I'm just thinking if you could actually distinguish between these two things, and I don't have a great answer for how exactly you would do it. Yeah, I'm gonna think more on that, Fran. Maybe we should chat about that.
No, it's just an interesting challenge because I've struggled with this as well. So I think it's a good question. So.
No, it's a good question. I
like,
don't want to just brush you off.
I'd like to jump in here. Sorry, Eva, but I'm the guy who thinks that there are paleosols in the cap carbonates, that the cap carbonates are largely calcareous-less. And there are also paleosols in the cryogenian. I found them in the Death Valley region. So that would certainly explain why the anomalies are high there. It's terrestrial, it's not marine.
I mean, from everything I've seen, I don't think I would follow that interpretation. Like I've been around, seen a lot of cap carbonates, and I don't think that makes sense to me. But it's something but I'll be honest, I haven't really had that idea in my head as I've been looking at these rocks. So.
Well, I have laid out my argument in several papers, but in the cryogenian case, we have pattern ground, periglacial pattern ground during the cryogenian at the same levels where you get the greatest anomaly, as Francis just pointed out. Anyway, it's an alternative idea that may get us out of this difficulty.
All right, let's move on to the next question. Thank you. So Eva Stuyken, please go ahead.
Wow, thanks for your talk. It was really great.
So
you showed in one of your last slides that the most important primary producers throughout the Proterozoic were cell bacteria. So I wonder if that's something that you just assumed, or does it actually fall out of your calculation somehow, that it has to be O2 production?
So this is an assumption largely built off of Joch and Brock's work. Okay. I know that there's there's obviously an issue a lot of people are grappling with find eukaryotic fossils and what exactly were they doing. But it was largely based on his records. Yeah.
Okay, thanks. And then so I have a second question, if I'm allowed. So there's a lot of talk these days about this new dark oxygen production. With oxygen being produced on these manganese oxides in the deep sea. And I know a lot of people are skeptical about this thing, but I've also heard that it's supposedly reproducible. So I just wondered if triple oxygen isotopes can actually just detect that signal, so it's if it's as a distinct signature.
I think that I'd have to get in touch with them, but I think the best person to ask about this would be Kevin Sutherland, who did his PhD work with Colleen Hansel and Scott Wonkel, and later postdoc with Dave Johnston. There's actually a lot, I think, of interesting work being done with clumped O2 and triple oxygen stuff in a range of environments, trying to constrain different reactions, seeing how they manifest and things we can actually measure. But the signal to noise is a lot smaller in the modern environment. And so constraining some of these things is challenging, and taking those insights and putting them back in the geologic record, I think is very, very hard. Yeah.
Okay. Yeah. Thanks.
Yeah.
All right. Thanks, Eva. Next, Lyle Nelson, please go ahead.
Thanks, Pete. Awesome talk. I wanted to go back. you might have gone through this, but I don't know if I understand totally. But you gave a percentage of modern oxygen of oxygen that goes into the sulfide reduction of a pretty precise percent of like 14 to 17%. I assume that's based on like modern experiments, and I was wondering, is that controlled for the percentage of, or the amount of oxygen in the atmosphere? Does that percentage change as you reduce the amount of oxygen? And then when you showed your facies dependent of like terrestrial to open marine, is that something where you think that's a change in the percentage of where your oxygen is coming from?
Yeah, so the results I said, it was from Belcher in 2007 paper. There's been subsequent work by Isaku Cole at Hui-Ming Bao's lab, slightly changed a little bit of that relationship, and a few other sulfide oxidation studies. In general, I think most of those find, a minority of oxygens in sulfate can be derived from O2 during pyrite oxidation. But when we think about specific depositional environments, I think it's not just that there's the initial oxidation, there's a whole suite of processes we need to take account for. And I'm not going to like stick my neck out and say exactly the proportion of everything, but that was sort of the thinking behind the study was like, let's just look where the biggest signal to noise is and see if there's any difference between things that we can confidently assign an environment, and then we can speculate on why that's the case. But if you're getting into things like the specific amount of O2 at the site of pyrite oxidation, that versus the degree of microbial sulfur cycling or other reactions that occur in the subsurface, parsing that out is not something that we're there yet. Does that answer your question?
Sort of. I was just wondering, in the calculations, do you like assume like a reduced percent? Do you always put
yeah.
or you just like reduce to put a range in or something like this?
No, exactly. So in previous works, that's been the way to do it, is constrain the amount of oxygen incorporation into sulfate. But there's clearly an environmental dilution factor that kind of permeates these data sets. But what that value is and how we should properly assign it to different environments, that's something that's requires a little quite a bit of thought and thinking. Yeah.
Cool. Thanks.
All right. So that there are no more questions or hands up at the moment. So I'll give, there's some time for someone to speak up if they want.
So maybe I ask Peter, great talk. So I have two questions, maybe I separate them. So removal of terrestrial productivity, I think to me, it's not so straightforward, because if you remove plants from land, you might be delivering more nutrients to the oceans. So actually productivity in ocean might increase. On the other hand, plants on land has effect in terms of producing more clay and enhancing weathering. So I'm wondering, what's your thoughts in terms of how removing plants on land could affect and potentially increase or decrease productivity in the ocean?
Yeah, so, I think my intuitions on this were sort of wrong. Like looking at recent work out of ice core records, when you, for example, during the last glaciation, it looks like marine productivity spun up a bit, and you didn't see a large decline, and in some instances, I think maybe a slight increase. So that's important. But thinking about all of the feedbacks that might be at play when we devegetate the whole terrestrial biosphere, and how that might play into different O2 levels and things, it's challenging to, it's really, really hard to be confident in, a general case of, if you remove all life on land, exactly what does that do to marine productivity? That's tough to say. There's a lot of like interesting feedbacks and relationships there that people study, and how that all fits together. I think it's a tough question.
Thank you. And second question about Belcher record and sort of recovery from overshoot. I obviously like your interpretation, but I'm wondering, did you consider effect of CO2 or like what potentially could be contribution of, let's say, increase or decrease of CO2 at the same time?
Yeah, so this is where we relied on estimates of CO2 from both modeling, so studies, people like Wolf and Toon, Paleosol work, folks like Nathan Sheldon, which I know these records have, a lot of uncertainty as well, but so we put in the CO2 constraints of different time intervals to derive our results from. If we held CO2 constant, I could take a look at that, but I think that even if you did that with the O2 as well, and I don't think that we inferred a huge CO2 change over that time, you still require a shift in productivity.
Yeah, I don't think it's a problem, but we, during GOE or during Lamagundi time, we typically see very enhanced chemical weathering as well as organic carbon burial. So it's actually a puzzle why it didn't go, CO2 didn't go to very low values. And I don't know what exactly happened with, to CO2 after Lamagundi event, but if anything, I would suspect maybe it would go down. But on the other hand, maybe with less burial with organic carbon, it could somewhat recover it. So it's still kind of open question.
Well, thanks.
Thanks.
All right, great. Thanks. So I'll just leave a few more seconds. If anybody wants to speak up and ask another question, feel free. And next week, we have, who's talking next week? I think you contacted us. Yeah, it's Mark Laflamme.
Oh, right. Okay. So we'll stay on the current stuff. Yeah.
All right. So thanks a bunch, Peter. Good talk and lots of great discussion at the end there.
Yeah. Thank you all so much for attending, and thanks again for the invitation. Really appreciate it.
Yeah. All right, everyone. Take care, and we'll see you next week.